of many different professional societies, was the head of the biology directorate and a staff for 4 years. And I think that really speaks to his commitment to this field and his passion for birds and for ecophysiology, which is what he's really built an army of environmental endocrinologists. And his family tree, if you go on, I don't know if you've seen the neuroscience family tree, things, but his is enormous. And even more, I think, what speaks most, I think, about me to John's success as a career is how much his students adore him. Whenever we're at meetings, there's always a large crowd of people trying to talk to John, and it's mostly his enormous family, his scientific family. So without further ado, he's here to talk to us about weathering the storm, challenges faced by migrant songbirds in the Arctic. And I will just make one quick plug. Tomorrow at 4 p.m., Marilyn Romanowski will be giving a talk about migration in Johnson 102. So we're lucky to get two wonderful talks for this opening of our seminar series. Also a social this evening at my house, and I can, if anybody here is interested, I can give the address, and that's also been sent around to the various listservs. So hopefully some of you can swing by. We're going to burn a lot of stuff. All right, John.
Sorry.
So people comfortable if I take this off? Thank you. Well, thanks, Jamie, for a very generous and kind introduction. Feels kind of strange the first time in 2 years I've been doing this, standing up here in front of an audience with a screen projection. So it's good to be back in the groove, so to speak. What I'd like to talk about today is sort of the end point of a lot of field work that's been done around North America and the Arctic. And talking about academic family, I just want to show a few. This is a long-term study, and these are the names of some of the key players. It's not complete, I couldn't get them all on there. But I just wanted to say this is much their story as mine. They had tremendous input on what I'm going to talk about today. And weathering the storm, challenges faced by migrant songbirds in the Arctic. We all know about climate change and how things are changing rapidly, and they're changing most rapidly at the poles. And over the years, we've been able to get some perspective on what actually is happening to Arctic songbirds in a very severe environment and what the future might hold. But in doing that, I'm going to muddy the waters a little bit, because I think we've reached a turning point where we have to take a rather different approach. So see what you think about that. It seemed to have frozen. That one, okay.
Well, I don't know, maybe.
I have many choices here. Okay. So a changing world, a changing perspective. Sorry, I don't mean to be hiding behind this. Oops. I'll get this right in a bit. We all know that seasons are shifting with climate change. Some of them are longer and warmer, others are shorter. There was a recent report that the South Pole recorded its coldest temperature ever. So it's not all a question of warming. We also know that weather perturbations, weather events, extreme weather events are becoming more frequent, of greater duration and intensity. Although not all individuals are exposed to changing environments in the same way, there are differences according to sex, territory quality, predator presence, and so forth. So these can all influence how individuals respond to exposure. And there's also a need to integrate daily and seasonal routines and exposure to these perturbations specific to individual experience. It's what we call the developing concept of the exposure, which is unique to the individual. I'm going to focus today more on perturbations, particularly the perturbations of weather. Sometimes it's called stress. I don't want to go into that because I think stress is a very misleading term. But it does involve two major processes. I just want to mention these quickly. There are the behavioral responses that animals use to cope with environmental perturbation. This includes the fight or flight response, what we call the emergency life history stage, changes in metabolism and immune system and so forth. Facultative migrations, when animals are forced to leave an area because of a perturbation. Reactive, proactive coping styles, sickness behavior, and others. Then there are the physiological responses, which are two major groups. First, the cellular responses, the heat shock proteins, DNA repair, mitigation of oxidative damage. It's a huge field in itself. The humoral responses, the hypothalamo-pituitary-adrenal axis, which I'll be talking a lot about today, and the glucocorticoids, that are the end product of that axis, adrenal medulla, the autonomic nervous system, epinephrine, prolactin, many other hormones, including the cytokines and the immune system. So, weathering the storm. First question might be, do Arctic blizzards cause repeatable changes in stress physiology and body condition in breeding songbirds? So that's what I'll next. But first of all, just a quick introduction to what the songbirds are and what we have been studying. And we've been working, my group's been working up at Tulip Lake Field Station on the North Slope of Alaska, run by University of Alaska Fairbanks and the National Science Foundation. And this is right alongside the let's walk over here. Here is a large portion. You see the Tulip Lake Field Station is actually an old pipeline camp that runs along the Dalton Highway and the pipeline as well. It is now much more modern. It's an excellent place to work. One of the advantages of this Tulip Field Station is the Dalton Highway. It's pretty much north-south, means we can get to areas of perturbation at different latitudes in this area very, very quickly. I don't know of any other place in the Arctic where you can do that without a helicopter or something like that. Here's the field station itself. There are many facilities here that we can use for studying the physiology of animals in the field. And also a lot of our birds are right there around the field station. And these birds are, there are two that I'm going to focus on today. One is the Lapland longspur, also called a Lapland bunting, which is an Arctic specialist, a whole Arctic specialist. And Gambel's white-crowned sparrow, which is a migrator, long-distance migrant, winters in the western and southwestern United States, and breeds throughout the northern boreal forest region, and including Arctic Alaska. And it's not a specialist in the Arctic per se, but it does get up there. So interesting comparison. Another interesting comparison about these two is that the Lapland longspur breeds builds their nests out on the open tundra, the tussock tundra, and so forth. And the white-crowned sparrow breeds in the shrubs and so forth along rivers and also other areas. These shrubs now are getting 2, 3, 4 meters high. So the white-crowned like this. Work done with the collaboration with Natalie Bolver at Columbia University. If we look at the number of nests of white-crowned sparrow in black and the Lapland longspur open bars, the Lapland longspurs quite clearly prefer the taller vegetation height, the shorter vegetation height of the tundra, whereas the white-crowned sparrows are preferring the higher shrubs. They nest inside, usually on the ground. If we look at the type of tundra too, there's moss, rhamnoids, or shrubs, see that again there's almost complete separation between white-crowned sparrows, mostly preferring the shrubs, and the Lapland longspurs out in the grasses and moss in the tundra. So an interesting comparison right there. They're in two different habitats on the tundra. This work again done by Natalie at Columbia shows modeling a random forest model. The top there, that is the present day. The green is the Lapland longspur habitat. The blue is the Gambel's white-crowned sparrow habitat. We don't have much for the North Slope, just simply because the satellite data don't do much really above about 65 degrees north. But the predictions are using the various models. On in 2050, assuming 5-kilometer dispersal of the lands and so forth, about 22, the Gambel's white-crowned sparrow habitat will increase by about 22%. The Lapland longspur will decrease by 18%. If you look at the limited dispersal in the same year, the white-crowned sparrow habitat will have increased by 15%, and the Lapland longspur habitat decreased by 56%. So what we have then is one songbird, the white-crowned, that has the potential to expand its range in the Arctic, and the Lapland longspur is retreating. We have some data for the white-crowned sparrow. This is the Dalton Highway. Here is Tulip Lake, Prudhoe Bay on the Arctic Ocean. And in 1995, in the earlier days of our work up there, white-crowns were pretty much limited to the Department of Transport station here. Here's Tulip Lake. And there weren't really many, if any, breeding birds north of that. And even at Tulip Lake and these other sites, the numbers of white-crowned sparrows that would be present would vary from year to year, depending upon snow cover and how long it took to melt off. So in some years at Tulip, there'd be just one singing male. And the next year, there'd be 40 pairs. So there's a lot of movement around there. By 2005, we'd already begin to notice the shrubs were getting higher. The white-crowns moved north to a place called Ice Cut in Happy Valley, where there'd been none before. They were now breeding. Then in 2011 to 2013, they got even further north, about 150 kilometers overall from Tulip Lake to Franklin Bluffs. And by now, I'm sure they're probably up on the Arctic Ocean, just from shrubs. So I don't want to spend much more time on that and what accompanies the changing range in these birds. But while the white-crowns are invading the north in the shrub areas, we do know that the Lapland longspurs are retreating from areas where they used to be. It's not easy for us to study them because if they're no longer there, we don't know if they died or whether they moved somewhere else. If they are invading the north, we can follow that. So having said that, with warming, the Arctic is changing in habitats, more shrubs, and they're getting higher. There are still storms that come through, mostly from the Arctic Ocean and also from Siberia. This is a typical example of a fine spring storm. And whiteouts are common, and they have to make sure they train the students and postdocs and so forth early in the season how to deal with whiteout. And this is not unusual. It's been known for a long time. And there's an old Finnish proverb, the northern summer is short and variable. My favorite quote is from the Sami reindeer people, probably, spring is the weather struggle time. That sums it up completely. How does winter doesn't want to let go, in other words. So I think spring is progressing and then boom, you have one of these storms come in. And these are little 25, 30 gram songbirds that have to cope with that kind of issue. Another thing is the variability from year to year. If we look here for a view on the Arctic National Wildlife Refuge, top two panels, the same day, in 2 different years, or more or less the same day. You can see in this flat plane here, the amount of snow cover is very, very different. So the birds don't often really know until they arrive in their breeding area what the conditions are going to be like. Even more interesting in the south, it's in the Brooks Range, Atherton Pass is one way that the pipeline goes up the pass, but also many migrant birds go over, and caribou go over that pass up into the North Slope. In 2002, again on the same day, the situation in the south is complete snow cover, and it's cold. And the some birds are moving up along this highway here and waiting to go over the top, but mostly they find refuge here and just wait until the snow clears. There's no way that I can see that they could realize that in that year, if they'd just taken the 40-minute flight over the pass, they would be clear. Go on. So there's a lot of unpredictability. The birds have to be very flexible for local conditions. So that just gives you an idea of some of the environments. Snow depth, the top, and temperatures are missed. So I'm not going to talk much about it. It just varies dramatically. One day it can be +10, the next day it can be -20, even in May and June. At the top there you see snow cover. Date here in 4 different years. The main point is at the top there, LA is the average arrival of black and monsters, and E is average egg laying time. And this is over all the years. The white crowned sparrows arrive usually a little bit later, not always, but egg laying is about the same time. 2013, which is the solid line with squares. That was the snowiest year, and then the warmest year, I think, was 2014, the next year. But everything else in between. The birds don't know until they arrive how deep that snow is going to be and how long it's going to take to thaw out. Here is just a summary of mean arrival time, which is the closed boxes there. The open box is the earliest arrival, and then A is the mean first egg laying date, and then mean egg laying dates again. You see in the blue is 2013, that very snowy year, so arrival and egg laying are certainly arrival is delayed, settlement compared with the other years. Look here to look here at 2013, again in the blue, egg laying dates are greatly retarded. This is the proportion of not laying eggs. So beginning here, nobody's laying eggs. Then as they start, more and more of the population are laying eggs till they're all completed clutches. With the Lapland monsters, the 2013, that snowy year, was the latest year that we've seen. And with white crowns, it was even more. So the white crowns were spread is even more. Main point again here is just that every year is different. And egg laying dates can vary over a restricted range because they have to get on with it. They can only last, the breeding season only lasts about 4 to 6 weeks, although that is getting longer. And the phenology also suggests that arrival times are very different, although egg laying times are more synchronous, at least with the monsters. So one final point to make. Again, here's the snow in 2013 and the others. The main point here is see all these changes in snow depth. These are storms that come through in spring. Some are even late. So this one happened with incubating eggs. We once had two feet of snow on July 4th. So these things can happen at any time. But this is the period when you think, okay, the snow's melting off, and then boom, you get these storms come through. And just to give you an example with that storm later, when birds were incubating, the storms here, and they flock. So here's a big flock of black-necked lobsters. We have traps set out with the bait in the sea, so obviously we come to that because there's some food there. But a lot of these birds are in really bad shape, and some of them die. So this is a big deal. So we get back to hypothalamic pituitary adrenal axis and responses glucocorticoids to these kinds of events. Just want to summarize first, what is a glucocorticoid response to motivation such as a storm? I'm not going to show you the actual experimental data because this goes back 50 years or more, but we also it's aggressive.
I have it fixed. I'm sorry, nobody on Zoom can hear us. So. Sorry. I just have messages coming in.
So, everybody can hear me better now. I'm not used to talking all day after a year and a half in pandemic, so my voice is getting a bit croaky. Okay, they suppress unnecessary physiological and behavioral functions such as breeding. They activate facultative behavioral patterns that promote survival, temporary emergency behavior, part of the emergency life history stage. Prepare the immune system for possible infection and wounding, sickness behavior, rather than inhibit it. Increase gluconeogenesis for combat to cold and also for local movements. And these taken together allow the animal to avoid the long-term effects of stress-induced high levels of glucocorticoids. So they really are trying, in a sense, of providing coping mechanisms for the animal to avoid chronic stress and potentially death, although that does happen. How do we study this? Well, if we capture the birds in those traps, for example, or in nets, and we collect a blood sample as soon as possible after capture, say within 2 to 3 minutes, we can use the corticosterone level in that time as a baseline, closest to what we might expect the bird to show before capture. And then the acute stress of handling and restraint, captured more samples taken in 5 minutes, 10 minutes, 30 minutes, and 60. They show this typical stress response of an increase in court that then maxes out at what we call a stress level. And this is common throughout the vertebrates, including humans. This is very, very typical. If we then plot the baseline levels as a function of time of day, we don't see anything like we do at the 30-minute and 60-minute levels, which suggests this is not just a circadian change in corticosterone levels, but is actually induced by capture, handling, and restraint. So this is what we call the adrenocortical response to stress, perturbation, to acute perturbations, or the stress series. Again, I don't like the word stress, but it's not a whole lot we can do about that. The nice thing about this is that you can get, this is a standard way of stressing all of these animals. You can get the baseline, you can get the maximum level generated, you've got the rate of increase, and then you've got the area under the curve, which is a measure of how much glucocorticoid total the animal has been exposed to over that time period. All of these can be used to look at the response in terms of ecological context and so forth. So here we've got an example of these stress series for male white-crowned sparrows that are the long-distance migrants, the Arctic birds. And what you see is something we'd never expected. There's tremendous modulation of the stress response with time of year. So the red shows arrival at Toolik Lake, for example, with the greatest response, much less variation in baseline, mostly also late migration in dark blue, incubation in green, feeding of young in blue, and then the winter and molting times tend to be the lowest. Huge differences. So in these males, as they move north and arrive in the Arctic, they're actually increasing their response to stress. That's not what we expected. We thought they would become resistant. Females do the same thing, not quite as exaggerated as in the males, but clearly the same thing. In contrast, the Lapland longspurs, if you look at a comparison here of males and females early in the season migrating, early breeding, males and females, there's no significant difference in their response at all. And if we look also in the molting, non-molting animals, late breeding versus molting, there is a significant difference, but nowhere near as dramatic as it is in the white crown. So the white crowns are modulating their stress response as they come north, and the Lapland longspurs seem to be much less variable. Okay. If we look also at the stress responses in male Lapland longspurs at the top and male and female white crowns on the bottom, as a function of year, in the Lapland longspurs, you see there are variations from year to year. With 2013, again, the snowy year, the birds showing the greatest response compared with other years. The white crowns, less so, much less so, and not significantly so. The main points here is what we're looking at here are changes in fat on the top, muscle profile, size of the muscle in the middle, and body weight, body mass on the bottom. The main take-home points here, males in dark, females open columns. The main point here is that in 2013, birds the males all tended to show a decrease in body mass and body weight, females less so. So there is an effect in body condition too. If we also then look at stress responses as a function of the duration of the storm, day 1, day 2, day 3, then, so there's no storm, which is the control of the boxes, storm day 1, storm day 2, storm day 3, and then 1 day post-storm. Really, again, up here with the triangles and so forth, there's no consistent pattern here from within the storm. It probably is a function of body condition and so forth in both males and females. But the main take home there is there's a lot of variation that we can't explain as a function of the day of the storm, how long they've been exposed to it. These are Lapland longspurs, males and females. Again, if we go to the white crowns, much less variation. Again, a difference between the species. So to summarize so far, the glucocorticoid responses to weather in Arctic sunbirds. Severe weather events can cause rapid increases in HPA activity. The hormonal profiles can vary by species, sex, and year, depending on ecological and environmental context. Responses to severe events were repeatable in some cases from year to year, but not in terms of duration of the storm. This is likely adaptive for helping these organisms cope with perturbations of their environment. The question next is, how is the hypothalamo-pituitary adrenal axis modulated to enable flexibility in response to extreme weather? And are there common mechanisms underlying flexibility, or is each scenario different, even though the outcome appears to be the same? What I'm going to do now is focus mostly on the hypothalamo-pituitary adrenal axis in white-crowned sparrows, because that's the most complete story we have. At the moment, we have a collaboration with Simon Medal at the Roslin Institute at the University of Edinburgh, where we're doing a lot of transcriptome work and a lot of PCR for specific genes in up to 11 different tissues. So there's a lot going on, but we have a fairly complete story for the white-crowned sparrow. So I'm going to show you that. That's going to make a point about muddying the waters a bit even more. We start out with the hypothalamus pituitary adrenal cortex axis. There are various peptides that form a cascade from hippocampus input from perception of the environment through to release of ACTH into the blood, and this stimulates enzyme conversion of cholesterol to corticosterone, which is then secreted into the blood. It circulates in the blood. You get negative feedback, classic negative feedback mechanisms, which sort of act as a control that the whole system doesn't get out of control. There is also a transport protein, corticosterone binding globulin, formed in the liver that forms a carrier. And there's mediators of hypothalamo-pituitary adrenal cortex axis and sites of modulation that you could then modulate the secretion cascades at multiple points, increase the response or decrease it. You could also change levels of CBG expression in the blood, the transport protein, because it is generally agreed that once the hormone is bound to corticosteroid binding globulin, it generally does not enter the target cell. It's only the unbound, the free in the blood that enters the cell. So regulating CBG is another method. And then changing negative feedback. But then one other, at the time, was fairly unexplored component was the response of the target cells. The target cell responds to corticosterone coming into the cell, which can be modulated here and here. The target cells could also do that. So here's the target cell, HPA axis. So once corticosterone has got to the target cell, it can interact with genomic receptors, the mineralocorticoid receptor, low affinity, saturated at low levels of CORT, and the glucocorticoid receptor that is saturated, is lower affinity and saturated at higher levels of CORT. There's also a non-genomic receptor. We'll talk a little bit about that, which mediates very rapid cell responses. That is another thing that could be modulated. After the corticosterone binds to the genomic receptors, it becomes a gene transcription factor, and there are co-repressors and co-activators that can influence that too. There are two enzymes, actually three, but the third we don't know what it does yet, but there are two enzymes called hydroxysteroid dehydrogenases, HSDs. HSD1 will promote glucocorticoid binding to its receptor, so it prevents its degradation, so it persists longer and more likely to interact with the receptor, whereas HSD2 deactivates corticosterone. It's a deactivation shunt. So those two do very different things. So I know you look at this, I mean, it scares me half to death too. It's very complicated. But, you know, I think going on from now into the next stages, we've got to deal with this somehow. We've got to come to grips with it, because it's quite clear. Tweaking any one of these points here could influence the final response, whether it's metabolic, behavioral, what have you. Okay. So what I'd like to do now is just spend the next few minutes just telling you about what we know about these target cell responses. And this means that the target cell can respond in very different ways, whether or not the corticosteroid level is changing. Yikes, we never really appreciated that before. And these target cells could also modulate themselves, their responses to cope very differently, whether it's a liver target cell or a neuron, or adipose tissue, or gonad. We have about 11 tissues we're looking at. I mentioned that the transcriptomic analyses of target tissues are currently underway. This is for white-crowned sparrow. It's going to grow, we know that. And we also have a lot of data coming now for Lapland longspurs. So we'll be able to make the species comparisons under similar contexts. Start with CBG. Produced in the liver. And this is work we did quite some time ago. Michael Romero, when he was in my lab, did a lot of this. And the black bar here is the CBG. You can see at the breeding, early breeding, in particular, CBG levels are much higher than they are in winter and during migration. This just again reminds us of the baseline corticosteroid level and the enhanced response to stress in these males compared with winter and migration. So it's thought that CBG might be playing some sort of protective role in protecting the organs from very high levels of corticosteroid. The question of why it would do that, we'll come back to that in a minute. Kriya Brunner, when she was postdoc in Texas, did some interesting work. She came back, she was a graduate student in the lab, but then came back for a little while to work on this. They looked at the specific binding, the actual biophysics of binding to receptors in birds from Alaska versus Seattle, the non-Arctic, so mid latitude bird. And CBG binding in the blood was higher, as Michael Romero had found, in the Alaska birds versus Seattle. And if you looked at the amount of free corticosteroid in the blood as a function of latitude here, blue being Alaska and the shaded bar being Seattle, at baseline levels, there was really no difference in the amount of free corticosteroid. But after 30 minutes of stress with increasing levels of corticosteroid, we found that the free levels increased in Seattle, but very much less so in Alaska. Completely the opposite of the total level in the blood. The way she was able to do that is because with the binding, she knew what the affinity of for binding corticosteroid was of CBG. She knows what the total plasma level is, and using an equation, she could work out how much would be bound to CBG and how much would be unbound, free, and able to enter the cells. So here, this is completely different from what we would expect from the total blood levels in Alaska. The free levels are greatly reduced compared with other latitudes. We also know that gene expression in the liver for CBG changes very rapidly. And if we look at our stress series again, but this time looking at the brains, livers of these birds, sorry, at 3 minutes post-capture, 10, 30, and 60, you see there is a significant increase in CBG expression. So CBG expression and release into the blood can change very rapidly and then provide this protective role. But what we also know is, this is work done by Sharon Lin when she was a graduate student in the lab. If you food restrict birds for 24 hours or so to mimic one of the aspects of a storm. The snow covers their food. What we found was that CBG, specific binding, decreased in the fasted birds versus the controls. So now it's going down the other way. Total corticosterone level in control and fasted birds was not different, which was a bit of a surprise to us. But the real surprise too was that if you look at the free corticosterone level calculated by her, the fasted birds showed an increase in free corticosterone versus controls. So early perturbation increases gene expression of CBG. You would expect it to play initially a protective role, but if the stress persists, the perturbation persists, as shown by this fasting experiment, then CBG levels are going down and there's a concomitant increase in free cort, which is able to enter target cells and trigger responses. So if we look at these genomic receptors in Alaska versus Seattle white crowns, if we look at the mineralocorticoid receptor, this has a lower affinity, I beg your pardon, a higher affinity, and is saturated at lower levels of corticosterone because of the high affinity. No difference, Seattle and Alaska, in the liver, but in the brain, there are many more in the mid-latitude birds than they are the Arctic birds, which suggests a possible resistance modulation. But if you look at the glucocorticoid type receptor, lower affinity, and is saturated only at higher concentrations of cort, this has been called the stress receptor in many cases. What we see here again is no difference in the liver with GR, but and no significant difference in the brain either, but a tendency like we see with MR in the Seattle, the mid-latitude birds. Continuing Kriya's findings here, she looked at the glucocorticoid type receptors that would be occupied at 30-minute free corticosterone levels. And the reason she can do this is she knows the affinity of these receptors in CBG, affinity of binding, and she knows the total concentration of corticosteroids and the free, what would be entering the cell. She can then calculate the nanomoles of corticosterone bound for milligrams of protein in the mid-latitude Seattle versus Arctic Alaska birds. And in the liver, the glucocorticoid receptors were occupied far more than they were in the Alaska birds, and the same was true in the brain. So although it looks as though we're bouncing back and forth here between the receptors in the birds or the cort and the CBG in the birds in the Arctic are increased, but they seem to be protected by CBG. But the numbers of receptors and the amount of hormone bound can actually be different depending on how you work these out. So what this led to is the corticosterone binding globulin, or the globulin buffer hypothesis, that the adrenocortical response to acute stress is enhanced at arrival, then blunted as breeding progresses. This modulation may have ecological basis that we know a lot more about now. There's an androgen-dependent, I didn't mention this has been shown in juncos and also chickens. There's an androgen-dependent increase in CBG. So androgens act on the liver to increase CBG transcription and blood levels. And this may buffer stress-induced increases so that the response of free corticosterone may be actually reduced. And Kriya's work actually showed that's what appears to be happening. So it is acting as a buffer. If the stress and perturbation persists, then within 24 hours, CBG corticosteroid binding globulin levels decline, thus increasing free corticosteroid levels that can enter liver cells, target cells in the brain, and so forth, and trigger a greater response. So by modulating CBG, you can have all sorts of outcomes downstream. Going on to those two enzymes, 11-beta HSDs. First of all, we didn't really know. We know the genes are in the genome of these songbirds, but we didn't know to what extent they might be expressed and whether they had the same actions as they do in mammals, for example. So another student in our lab, Jonathan Perez, used blockers of HSD1 and HSD2. And metyrapone, the triangles there, are blockers of HSD1. Should decrease corticosteroid. They do in males, but not so much in females. DTC is an inhibitor of HSD2, the deactivation shunt that should then increase CORT levels, which is the circles there, with the squares being the controls in between. So, and that was also true of females. So what Jonathan was able to show was that, yes, it does look like HSD is in cells and is responding with corticosteroid release in the blood and also in tissues, as we would expect from what we know it does in mammals and chickens. So with that in mind, we then started to look at gene expression of HSD1, which enhances corticosteroid presence in target cells, males and females. You see in the liver here, it is high expression, higher in male than in female, less so in the kidney and the adrenal gland and gonad, and not very much in the other tissues, which was a bit of a surprise. HSD2, almost none in the liver. It was more in the adrenal gland and kidney, and less so in the other tissues. We hadn't really expected that HSD expression would be so limited to so few tissues. And there are various ways one could interpret that. And we're also now looking in Lapland long spurs, the same genes, and same tissues. So hopefully we'll get some data on those very soon. And this again is one of these complicated graphs, but this is just looking at gene expression of 11-beta HSD at the top and 11-beta HSD2 in hippocampus, hypothalamus, pituitary, and also the liver. No change. So there's not much more to say about that. There's no change over restraint time, rapid change to a perturbation, unlike CBG. So it is a player. The HSDs are a player, but it probably in far fewer tissues than we thought. So that's good to know. Just to remind you here that again, that the comparing parental and pre-parental early breeding and later breeding, the corticosteroid levels are increasing in earlier, showing this enhanced response earlier in the season versus later in breeding. Because now we're going to look at GR and MR expression. GR and MR are expressed throughout the brain, as we would expect. These are coronal sections, more anterior, medial, and more to the rear. The main point is these are MR on the left-hand side, GR on the right-hand side. And we're going to focus now mostly looking at the hypothalamus. We've been cut out of the brain to look at gene expression, and the hippocampus, which is right, if you look at this black bar, it's right at the top there. where there's quite a lot of expression of GR and MR. Reasons for that are that the hippocampus appears to be an important starting point of this whole stress or perturbation cascade. So it appears to be regulating hypothalamic secretion of peptides that then trigger that whole cascade. And if we look at GR to begin with, no differences in the hippocampus or the here you can see the GR localized mostly in the paraventricular nucleus, which is where a lot of the neurons are that release peptides in this cascade. No difference, and this has been repeated by many others too, that GR expression seems to be fairly level, kept constant. With MR in the hippocampus, there is a difference in both from comparing pre-parental and parental, early breeding and later breeding. And this seems to be fairly constant as well. So the GR are being maintained at fairly constant level and MR less so. If we look at GR and MR changes during the acute stress, same tissues, GR top, MR bottom, no differences, no significant differences. So we don't think there's any rapid change. But the only one that appears to be modulated so far in any tissue that we've looked at is the mineralocorticoid receptor, MR. And then finally, we know that there are very rapid responses to some of the steroid hormones. Genomic receptors take, down fast says, at least 30 minutes to see any mRNA. Mostly it's hours, and certainly downstream any protein effects takes hours. But we do see responses, behavioral responses within minutes of a surge of corticosteroid. So Kriya Brunner again did this as part of her thesis in the lab. One of the problems is looking at this just by capturing the animal, capture, handling, and restraint increases corticosteroid. So how do you control for that? Well, what she figured out was that these birds really like mealworms. And we could spike the mealworms with corticosteroids in oil or DMSO, dimethyl sulfoxide. And the birds would then eat the mealworm and ingest the corticosteroid without us intervening at all. And when she did this, what she found was then she would take blood samples at intervals and found that if you look here at corticosteroid levels minutes after the mealworm was ingested, that at both the low dose and the high dose, there was a peak in the blood at about 7 minutes, going on through 15, and with the high dose after 30 minutes before it was all cleared by 60 minutes. So yes, it was very successful in introducing a non-invasive way of introducing corticosteroid that we could then look at behavior. And the behavioral data showed that if you look at perch hopping activity associated with trying to move away from the perturbation, which is typical response, behavioral response, she got a response at the with 4 micrograms of cort in DMSO within 15 minutes of ingestion. And that persisted out to 30, 45 minutes, whereas uninjected mealworm and mealworms with DMSO alone showed no such response. So this suggests that there is a rapid response receptor, non-genomic. And we don't know much more about that than that, but it is there. And furthermore, the final point is, it is modulated seasonally as well. So on long days, which induces migration and gonad maturation in these birds, the corticosteroid increased relative activity, whereas on short days, the same treatment did not. So what I wanted to point out here is that this is far more complicated than we ever imagined. You know, I started out working out ways to measure blood levels of steroids. And at the time that was, whoa, that was great. Now we know what the blood levels are. But now we're looking at all these other components: weathering the storm, coping with environmental change. Arctic songbirds face challenges of extreme weather when they arrive to breed. The flexibility in physiology, morphology, and behavior is needed to cope. The hypothalamo-pituitary adrenal axis and target cells in the brain, liver, gonad, and others provide networks for coping mechanisms, we should say potential networks. There are multiple sites in this axis where behavior and physiology could be regulated in response to weather. Target tissues may be able to adjust their responses to corticosteroid, even though plasma levels do not change or are volatile. And in the future, we have with transcriptomic analysis and quantitative PCR, we have 9 different genes already in 11 different tissues. Massive undertaking with my collaborator, Simon Medal at Edinburgh. And we will undoubtedly find more genes that are being greatly upregulated during these different stress scenarios and how they fit in. So I think a big challenge is going to be analysis of the data. We still need a lot of field context. So we have to bear in mind what the birds are doing at the time. And discriminate function analysis, path analysis may also allow identification of stress phenotypes across seasons among individuals and how they may cope with change. So we're at this point where we're just sort of scratching our heads. I think, you know, Suzanne Austin has dealt with this with parental behavior in pigeons. Meta Landis also dealt with some of this when she was in the lab. Okay, and Jamie too has started to look at GR and MR and other things. So there are other people working on this across North America and Europe as well. So hopefully as these techniques evolve and bioinformatics analyses allow us to do more things, we'll be able to sort out this sort of tangle. It's up to you guys, the young people, to take this forward. I'm done for now. I look forward to seeing the data and joining in discussions of interpretation, but it's a great challenge. And, so hope I've given you some idea of so-called stress physiology and behavior and climate change, but didn't expect it to be this complicated. But we'll work it out. I have every confidence. Thank you.
So thanks for that, John. That was interesting. The story does keep getting. I'm gonna put my Frank War hat on here since Frank is not here today with us. And so I can't remember. I'm sure I've talked to Maria about this, but the idea that you hinted at that the rapid effects of the corticosteroid that are not genetically, or at least don't look like they're having to do with nuclear, upregulation of corticosteroid, corticosteroid, production pathway. And I was really intrigued. You said the behavior there was that the bird started to, I guess, sort of flit around and so forth. As I recall, what Frank and Miles Orchinik were showing was that that rapid response was making the newts migrate, or it increased locomotor behavior. So is there anything else we have? I guess it's probably speculation now, but this whole, this whole idea that this kind of a non-nuclear effect or a sort of a membrane bound receptor for the
Yeah,
corticosteroids.
we assume it's membrane, but the steroids can cross membranes so easily and
Yeah.
rapidly. It could be an internal receptor. It's just not a genomic. It doesn't
Yeah.
form a gene transcription factor. Yeah, I was familiar with Frank's work, and Jacques Balthazar has just put out a paper on the history and scope of non-genomic receptors for hormones. I haven't had a chance to read it. Marilyn found it the other day. I haven't had a chance to read it yet, but I think there'd be a lot of insight in that. But we have data going back to the 90s showing how corticosteroids can trigger this kind of escape behavior. We call this facultative migration, getting away from an area where bad things are happening. And what Lee Astheimer found when he was working in the lab was that it's also context dependent. That if the birds had food in the cage, corticosteroid would trigger the opposite. They become completely quiet. But if you remove the food, then they were really trying to get out, because they're trying to, in the corners, they're trying to get their heads through the bars of the cage, and they're trying to get away. Same treatment.
Yeah.
But if they have food, it makes sense. Why leave?
Yep.
If you've got the food there, just ride it out.
Yeah.
But if you don't have the food, if that's suddenly gone, then you have to go to. How that's mediated, we don't know. Many of the unknowns. But yeah, that's really intriguing.
So I was thinking about CBG more while you were talking, and that it's just so interesting that if you're going to elevate court, why also elevate CBG if it's kind of keeping it from interacting with others? And it seems like one benefit would be that you've got this big supply of port in the bloodstream that's ready for the kid to be released. And if CBG is changing dynamically with handling stress, then maybe it does that with other things too, like maybe blood sugar levels or something.
Yeah.
Has anybody looked at that, like changes in blood physiology around port for CBG to know if it dumps port all of a sudden, if glucose levels drop or something like that?
No, I can't think of that specific. But initially when we got the hormone level data, we thought that this might be some coping mechanism, that if they can cope for a short time and then they leave, if the weather is bad. But then we found that they were doing that even when weather was not bad. It was just the timing of arrival. Then we found the CBG, as everything started to fit together. But beyond that, I don't think.
Because there's like the subset of people who are, the CBG as a delivery mechanism, and it's going to target, or not always target cells, but then maybe it's releasing cortisol at particular places. And I haven't kept up with that literature.
There is evidence that in mammals that some cells are able to bind CBG if it is bound to a steroid and internalize it. So it would be playing a transport role. It apparently does not bind CBG that is not bound to cortisol. I think it was liver. Yeah. And I thought about that and said, well, steroids pass through membranes so easily, why would you need to transport it? Unless within the cell, that CBG was transporting it to a specific receptor. I don't think there's been much more beyond that I know of. But it is an issue. That's why I'm looking forward to reading Jacques Balthazar's paper, because he might shed, well, he's very thorough, very complete. So if it's out there, it'll be in his paper. Yeah, those are interesting points. All of these things, we never really considered before. And I think with the genomics and the transcriptomics, once we identify all the major players, one thing we might be able to do is develop a chip, a microchip with all these genes on there that we could then test different tissues and see what combinations of genes are actually being expressed in the tissues. That's one possibility to allow to do that. But I've talked with the Roslin people about that, and they said, "Oh yeah, absolutely. If you're sure you've got the full repertoire of genes that are involved, we can do that." So that might help to sort out at least the context. Who knows where it's going to go? But something like that has to happen. Well, thanks everybody for putting up my croaky voice. I'm almost.